Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. Okay, so today we're going to talk about a new chapter in human physiology, that is excretory substance and their elimination. So this is chapter 4 in human physiology. So we are done with body fluids and circulation. So we are done with breathing and exchange of gas. Okay. So hello, so we have been talking about breathing and exchange of gas. We are done with digestion. So we are done with body fluids and circulation. So now we are going to talk about excretory substance and their elimination. Alright, yes, Antonia B, hello, hi, a very happy good evening. Yes, I'm good, thank you. How are you? Yes, Naveen, hello. So thank you so much for joining my session. So please do like, share and subscribe. Hi Leela, yes, a very happy good evening. So welcome to my session, and please don't forget to like, share and subscribe. So quickly share the link with your friends. So today we're going to talk about the most important part of your human physiology, that is excretory substance and their elimination. So ready all of you? Yes? So shall we start? So can we go forward? Okay, so let's go forward and see what we are going to talk about. So we're going to talk about the structure of kidney. So shall we move forward? Yes. So in this chapter, that is excretory substance and their elimination, so we are going to talk about the first thing, the human excretory system. So we are going to see what your excretory system is made up of. So how different organisms have different excretory system, right? Then we'll move forward with the topic urine formation. So what helps in urine formation and how is your urine formation done? Okay. The next thing is function of tubules. So you have various tubules which are part of your kidney, and they also play an equivalently important role. So we're going to talk about the function of these tubules as well. Then mechanism of concentration of filtrate. So this is very important, and this is where kids go wrong. So we'll talk about this as well. So yes, Ritu, a very happy good evening. So thank you for joining my lecture. So hi Munna, yes, hello. Yes, I'm good, thank you. So thank you so much for joining my session, Munna. So please do like, share and subscribe. Yes, Dharni, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay, so then we'll talk about who regulates the kidney function. So who is responsible for regulating the kidney function, and finally we'll talk about what is micturition. So micturition is the process of releasing urine, and finally we'll talk about the disorders of your circulatory system. Yes, Anthony, hello. Yes, I wished you first. Okay, right. Okay, so shall we talk about the human excretory system? Yes. Okay. So before we go into the human excretory system, so what is excretion? So why do we have to eliminate substances? So what are the substances we have to eliminate? So that is very important. So we have to talk about these things. So ammonia, urea and uric acid, they are the common forms of nitrogenous waste. So your waste things that you have to excrete are your nitrogenous waste. So your nitrogenous waste has to be excreted out. So what are the different nitrogenous waste that we have? So the different nitrogenous waste includes your ammonia, urea and your uric acid. Okay, so this includes ammonia, urea and uric acid. So these are the different nitrogenous waste. Hello Keshav, yes, welcome to my lecture. So guys, please do like, share and subscribe. Okay, so ammonia is the most toxic of all the nitrogenous waste, and large amounts of water are required for the elimination of ammonia. So the toxicity is in this order. The most toxic nitrogenous waste is your ammonia, so followed by urea and your uric acid. Okay, so ammonia is the most toxic, and for its elimination we require a lot of water. Okay, yes, Guha, hello, hi. So thank you for joining my lecture. So please do like, share and subscribe. Okay, so the uric acid is least toxic among these three, and it has less amount of water, and it needs less amount of water for its elimination, right? Okay, so this is the list of organisms on the basis of their nitrogenous waste. So if an organism excretes ammonia as its nitrogenous waste, so that organism is known as ammonotelic. So the excretory substance of this organism is ammonia. So it is ammonotelic. Yes, Shubham sir, a very happy good evening. So thank you for joining our lecture. Yes, Anthony, you're right. So infinity. So welcome to my lecture. So please do like, share and subscribe. Okay, fine. So, who are the organisms which eliminate ammonia? So, that is your aquatic amphibians and insects. So, they eliminate ammonia. So, ammonia is soluble and it is excreted by diffusion. Okay. So, their excretory product is ammonia. The next one is ureotelic. So, ureotelic, so their excretory substance is urea, right? So, it is terrestrial amphibians and marine fishes. So, urea is converted in the liver and it is released into the blood, filtered and excreted through kidneys. So, this is what happens in ureotelic. Then you have uricotelic. So, here the waste is uric acid. So, reptiles, birds and snails. So, uric acid in the form of pellets or paste. So, we belong to ureotelic, right? So, we are ureotelic. So, our excretory waste is your urea, right? So, you are asking me breakdown of tissues or in lungs. Okay, so it occurs in the lungs. Okay, Antonia B? Yes, okay, you are FF lovers, so after a very long time you are here, so thank you for joining my lecture. So please do like, share and subscribe. So thank you for joining my lecture today. I hope you are doing good, FF lovers. Yes, after a very long time you are here. So based on the excretory product, so these are the organisms. So ammonotelic, so they excrete ammonia. So ureotelic, they excrete urea, and uricotelic, they excrete your uric acid. Okay, yes. Oh, this YouTube blocked me from sending chats on that mail. Oh, okay, fine. Right. Okay, so please remember the examples, that is very important. So this is aquatic amphibians and insects. So this is terrestrial amphibians and marine fishes. This includes your reptiles, birds and your snails. Okay, yes. So you have to remember, so we have to excrete. So the excretory waste are mostly nitrogenous substances. So nitrogen containing substances. So what are the nitrogen containing substances we have in our body? So this includes your ammonia, urea and uric acid, right? So among them, so ammonia is most toxic of all, right? And this ammonia elimination requires lot of water. It requires large amounts of water. So this is least toxic and it requires less amounts of water. So based on this, organisms are classified. So they are classified as ammonotelic, then uricotelic, this is ureotelic and uricotelic. So here the waste product is ammonia, here it is urea and here it is uric acid. Okay, so these are mostly aquatic organisms. So we have them as aquatic amphibians and your aquatic insects. Okay, so then you have urea. So urea, it is by terrestrial amphibians and marine fishes. So please remember. Then uric acid is excreted by your reptiles and birds and to some extent insects as well. So your urea is converted in the lungs. So, okay, it is converted in the liver and then it is excreted out through the kidneys. Fine. Okay, ma'am, in evolution, the diagram about the timeline and origin and evolutionary species is important. You have to remember the era. You don't have to remember the individual species, but you have to remember the eras. In which era what came? So first which era came into being, then slowly what was the other era. So all these are important. Okay? Yes? Fine. So this is what we read in the first part. So this is based on the excretory substance. right? So now, what are the excretory structures? So how do we excrete the substance? So what are the structures that we have? So invertebrates have simple tubular forms of excretion. So vertebrates have complex tubular organs called as kidney. So this is invertebrates and this is vertebrates. So underdeveloped invertebrates have simple tubular forms and developed vertebrates have complex tubular organs. Okay? Right. So this is the structure. So protonephridia or flame cells is present in platyhelminthes and rotifers. Okay. So the role is they help in ionic and fluid volume regulation. Right. The next one is nephridia. So nephridia is present in earthworm. So this is in phylum Annelida. Okay. So the role is fluid and ionic balance. The next one is Malpighian tubules. So Malpighian tubules are present in insects like cockroach. So this is phylum Orthopoda. So they help in the removal of nitrogenous waste and osmoregulation. Right? The last one is antennal and green glands, which is present in crustaceans, which are class of your Orthopoda. Right? Yes. So this is normal excretion. So I will teach evolution on Monday. So it is like this: MWF I teach a class 12 chapter, TTS I teach a class 11 chapter. Okay, Leela? Yes? Okay. Okay, so you're asking about sodium. Okay, so can we talk about this when I'm talking? Okay, I will explain at the end of the lecture if you remind me. Okay, Antonia B? Yes? Fine. Is nephridia and protonephridia same? So you have three regions: proto, meta, and you have two types of nephridia: protonephridia and metanephridia. They are regions of the kidney. So at the time of embryonic development, so where do they arise from? So that makes this difference. Okay? Yes? Fine. So now is this clear? So protonephridia, platyhelminthes, nephridia in your earthworms, Malpighian tubules your insects, and you have crustaceans, right? Okay. So what did we read? So we are talking about your excretory structures. So how elimination happens with the help of excretory structures. Okay. So in case of invertebrates, we have simple tubular forms, right? So they help in excretion. So in case of vertebrates, we have well-developed complex structure known as kidney. So intermediate, how is the development? Okay. Hi Vishal. Yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Okay. So here we move forward. So in case of platyhelminthes, so we have what is known as flame cells. Right. The next one we talk about phylum Annelida. So this is where you have your earthworms. So they have nephridia. Then you go to phylum Orthopoda. This is basically your insects like cockroaches. So here you have your Malpighian tubules. Right? And then you have class crustaceans, so which includes your prawns. So they have antennal glands or green glands for excretion. Is this clear? Yes? Yes, Vishal, I'm good, thank you. How are you? So Anthony, have you joined the crash course? So Anthony, please ping me this doubt in the Telegram group. Alright, I will tell you in detail. Okay? Can we complete this chapter within today's class? No, because there are a lot of students who are watching this class for the first time. Maybe it is divided into 2 lectures. Okay, FS lovers? Yes? So this is the excretory structure. So excretory structures are also important, so because they might ask this in match the following. Alright Vishal, so thank you. Yes, human excretory system. So Vishal, how is your preparation going on? So now we're going to talk about the human excretory system. So the human excretory system has a pair of kidney. So we have a pair of ureter, we have a urinary bladder, and we have a urethra. Right? So we will start with the kidney. So kidney is a bean-shaped structure. Okay? So it is reddish brown in color. And what is its location? So let's talk about it. So we have a pair of kidneys. So first we'll begin with the kidneys. So what is the structure of kidneys? So this is very, very important. So we have a pair. Okay? So kidney is reddish brown in color, and we all know it is bean-shaped. Right? So where it is located, that is important. So it is located between the last thoracic and the third lumbar vertebrae. Right? And it is attached to the inner dorsal wall of the abdominal cavity. So this is the location of your kidney, right? This much is clear? Fine. So now what is the length? So the length of the kidney ranges from 10 to 12 centimeter, almost the size of your short size scale, okay? And the width is 5 to 7 centimeter. So the thickness, so I call it rho, so it is 2 to 3 centimeter. So the average weight of kidney is around 150 to 170 grams. Right? So now shall we talk about the structure of kidney? Okay, you also have registered the NEET application form. Oh, okay, you had registered. Okay, going through your notes and mentee. Okay, fine. Hi Shimpi. Yes, a very, very happy good evening. So yes, Shimpi, I am good. Thank you. So please do like, share and subscribe. Okay, you want me to teach biotech? Okay, fine. Okay, Anthony, because that is the suggestion you're giving me. Right. Okay, fine. Alright, Vishal. Okay. Yes, FF lovers, you're right. So that is the exact location. Perfect. Right. So this is your kidney description. So now moving into its structure. So how is the structure defined? So on the inner side of the kidney, so on the concave inner side of the kidney, so on the concave inner side, so you have a notch, you have an opening, so that is known as hilum. So it is through the hilum that your blood vessels, your ureters and your nerves enter the kidney. Right? Okay. So the outer layer of the kidney is covered by a tough capsule. So the outer layer is covered by a tough capsule, right? Okay. So the inner layer of your kidney is divided into two parts. So the first one is known as outer cortex and inner medulla. Right? Is this clear? Fine. So inner to the hilum. So inner to the hilum, you have funnel shaped structures which are known as renal pelvis. So renal pelvis has projections which are known as calyces. Is this clear? Anyone who has not understood till here? I am talking about the structure of kidney, right? So the kidney is located between the last thoracic and the third lumbar vertebrae. Fine. So your kidney has on the concave side, it has a notch which is known as hilum. It is through the hilum that your blood vessels, ureters and your nerves enter. Okay. Yes, Vishal, you're right. So the outer surface of the kidney is a tough capsule, and inner to the hilum you have funnel shaped structure which is known as renal pelvis, which has projections which is known as calyces, right? So the inner layer of the kidney is divided into the outer cortex and the inner medulla. So the medulla opens into conical mass which is known as medullary pyramids. Okay, so these medullary pyramids further project into calyces, they connect with these calyces, right? Okay, so the cortex extends through these medullary pyramids, okay, as renal columns. They extend through these renal pyramids as, through these medullary pyramids as renal columns, and these columns are known as columns of Burney. Clear? Yes? So, the inner layer of the kidney or the inner surface of the kidney is divided into outer cortex and inner medulla, right? So, the inner medulla is made up of a conical mass that is known as your medullary pyramid, which opens out into the calyces. Fine. So, the outer cortex, it extends through these medullary pyramids, right? So, these medullary pyramids, when the cortex extends through, they are known as renal columns. So, these renal columns are known as columns of Burney, right? Okay. So, this is the first part. Okay. So, now the next part. So, each kidney has nearly 1 million structures known as nephrons. 1 million structures which are known as nephrons. So, nephrons are the functional units of your kidney, right? So, nephrons are of two parts, that is glomerulus and your renal tubule. So, you have two parts of the nephrons. Is this clear to everyone? Yes? Yes? So, we go according to NCERT. So, don't confuse extra terms here. So, first let's understand the structure of kidney and then we'll go much in depth. Right? So, till this it's clear. Now, can we talk about glomerulus and renal tubule? Yes? I hope this is clear. So, we started from here. Then we spoke about the inner layer. So, how the inner layer moves we saw. Now, we went into a structure known as nephrons, right? So, nephrons are the structural and functional units of your kidney. Yes, so Shimpi has understood. Okay. So, how about Munna? Okay, so now I move forward further. Okay, so now let's talk about the structure of your nephrons. So, nephrons have two parts, right? So, one is glomerulus and the other one is renal tubule. So, first let's talk about glomerulus. So, glomerulus is a tuft of capillaries. So, what is tuft? So, tuft is a network of capillaries. So, capillaries are a branch of your blood vessels, right? So, they are a part of your blood vessels. So, here you have two arterioles in the glomerulus, okay? So, you have an afferent arteriole and you have an efferent arteriole. Fine. So, afferent arteriole brings blood inside the glomerulus. So, if this is my glomerulus, afferent arteriole becomes it brings blood inside the glomerulus, right? So, this afferent arteriole is a branch of your renal artery. Fine. Then efferent arteriole takes blood away from the glomerulus. So, this takes blood away and this takes blood to the glomerulus. So, you have two types of capillaries associated with your glomerulus, okay? Yes? Yes, Pallavi Dubey, that is Munna's name, new name. So she has come with that name. You took vaccine? No, but recently Kota is not having the stock of vaccine, so I have not yet taken. Yes, Satyendar, hello, very happy good evening. So welcome to my session. So please do like, share and subscribe. Alright, so here we move forward. So this much is clear. So this is your glomerulus, right? So now renal tubule. So renal tubule, it continues from a cup-shaped structure. So it continues, so we are talking about renal tubule. So renal tubule continues from a cup-shaped structure like this. So this cup-shaped structure is known as your Bowman's capsule. So Bowman's capsule holds your glomerulus. So this glomerulus plus Bowman's capsule is known as renal corpuscles, or it is known as your Malpighian tubules. Right? Okay. So Kalpita, yes, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So it's a very new name, Kalpita. So what does that mean? Does it have any unique meaning? Because I'm not aware. Okay, so anyways, thank you so much for joining our lecture today. So please do like, share and subscribe. So we are talking about the renal tubule now. Okay. Yes, so the glomerulus plus the Bowman's capsule is known as the renal corpuscle or the Malpighian tubules. Further down the renal tubule, it continues to form a highly coiled network which is known as your PCT, proximal convoluted tubule. Okay, yes, one day one chapter, so that is not yet been started. So now we are doing only syllabus wrap up. Okay, FF lovers. So okay, so Kalpita means imagination. Alright, so that's nice. Okay, fine. So proximal convoluted tubule. So after which you have an hairpin shaped loop, hairpin shaped structure which is called as your Henle's loop. Okay, so this Henle's loop has two structures, that is ascending and descending lymph. Okay, so you have two structures, ascending lymph and the descending lymph, right? So now the descending lymph further joins to form a highly coiled network, another highly coiled network, so that is known as DCT. So this is distal convoluted tubule, right? So this distal convoluted tubule, it opens into a straight tube that is known as your collecting duct. So collecting duct opens into the renal pelvis, if you remember, that is inner to the hilum, through medullary pyramids. Through medullary pyramids and calyxes. Is the structure of nephrons clear? Yes. Why the class timings have changed? Okay, now that your NEET exam dates have been announced, so I want you to give a lot of time for your self-study. So we were having two lectures, right? One from 4 to 5 and then from 6 to 7. So I cancelled off both and together we have one lecture that is from 5 to 6. So that you have time for your self-preparation. Okay, Satyendra? Yes? Fine. So is this clear to everyone? So how it started? So we are talking about the structure of nephrons here, right? In the morning, so morning I am completely packed, so morning I have so many classes, it is not possible for me to come in the morning, okay. So glomerulus and renal tubule, so glomerulus is a tuft of capillaries, so capillaries is a type of blood vessel, so you have two capillaries here, afferent arteriole and afferent arteriole, so this is a branch of your renal artery, okay. Then renal tubule, it opens from a cup-shaped structure. The cup-shaped structure is your Bowman's capsule which houses your glomerulus. So Bowman's capsule plus your glomerulus is known as renal corpuscle or your Malpighian tubule. Okay. Then from here it extends to form a highly convoluted structure that is known as PCT, proximal convoluted tubule. So this further forms your hairpin shaped structure, which is known as your Henle's loop. So Henle's loop has ascending limb and descending limb. So the descending limb further condenses to form a coil structure, which is known as DCT. Okay, so DCT opens into a straight tube, that is your collecting duct, right? So collecting duct finally opens into your renal pelvis through your medullary pyramids and calyxes. Is this clear to everyone? Yes? Yes? So is the structure of nephrons clear to everyone? Any confusion? So you should get the structure clear. Yes, any doubt you want me to explain again? Yes, I am ready to do, but ask me right now. Yes, quick. Yes, quick. No doubts? Can we proceed? Yes. Okay, Antonia B. So, Naveen, have you understood? Vishal, Darni, Shimpi, Leela. So, Swami, is it clear? No doubt? Yes. Shall we move further? Alright. So, now we move further from here. Yes. So, all these parts we have already spoken about, right? Okay. So, now this is the structure of your kidney. So, see, so this is your hilum. It is through the hilum that your blood vessels, your ureter, your lymph, your nerves enter your kidney. Okay? Yes. So, now inside to your hilum, so you have funnel shaped structures, okay, which is known as your renal pelvis. So, this has projections that is known as capillaries. So, this is your renal pelvis and this has projections which is known as your calyxes, right? So, the outer kidney is surrounded by a tough capsule. The inner surface is divided into cortex and medulla. So, this region is the cortex region and this is your medullary region. So, medulla has funnel shaped structures or conical structures which is known as your medullary pyramids, right? And they project inside the calyxes. If this is the calyx, you have your medullary pyramid here. So, it is structured like this. So, here I am showing you the practical picture. So, then we started talking about your nephron. So, here you have a cup shaped capillary. So, this is your Bowman's capsule which consists of your glomerulus. So, your Bowman's capsule plus your glomerulus is known as your Malpighian tubule or your renal corpuscles, right? So, this is the name and then it opens to form a highly coiled structure which is your PCT, that is proximal convoluted tubule. Then you have hairpin shaped structure which is known as your loop of Henle. So, this comes down, right? So, this is your ascending lymph and this moves up. So, this is your descending lymph. Then it opens into another structure which is your DCD. So, this opens into a straight tube, that is your collecting duct. So, collecting duct finally opens into your renal pelvis via your calyxes. Alright? Yes? Fine. Multiple question is going to come in NEET from this. So, you had a dream. Is that so, Satyendar? Okay, fine. So, this is from the structure point of view. So, now let me tell you something. So, you have your cup shaped structure. I am drawing the nephron. So, this is my this is my glomerulus, right? So, this is my afferent arteriole and this is my afferent arteriole. So, this is my Bowman's capsule. So, this is my glomerulus. So, together it is my renal corpuscle or my Malpighian tubule. So, this opens into a highly coiled structure and then we have loop of Henle, right? And then I have another coil structure here, so which further opens into my collecting duct, right? Yes. So, this is my PCT. So, these two are my loop of Henle. So, this is my ascending lymph. So, this is my descending lymph. So, this is my DCT, distal convoluted tubule, and this is my collecting duct. right? Okay. You can also take multiple question mock test practice for some classes. Okay, so you want me to discuss questions? Okay, your suggestion is taken. Okay, Satyendra, fine. So this is the structure of your nephron, right? So your Malpighian tubules plus your DCT plus your PCT, so everything is present in the cortex region of the kidney. So your PCT, your DCT, your Malpighian tubule, everything is present in the cortex region of the kidney. So the loop of Henle is present towards the medullary region, right? So this is the normal classification. But sometimes your loop of Henle, okay, I write like this. So loop of Henle sometime, what did I say? Okay. So this is descending lymph. Yes, you're right. So this is descending lymph because it is coming down, and this is ascending lymph. Okay, Leela? Yes? Okay, fine. So now your loop of Henle, right, when it is very short, it is also present in the cortex region itself. So when it is present in the cortex region, so those nephrons are known as cortical nephrons. So ideally your Malpighian tubules, your DCT, your PCT are present in the cortex region, okay, and your loop of Henle is present in the medullary region. But sometimes your loop of Henle is too short, so it is also present in the cortex region. So those nephrons are known as cortical nephrons. So if the loop of Henle is long enough and it extends into the medulla, so they are known as juxtamedullary nephrons. Okay, so we have very few juxtamedullary nephrons. Is this clear? So you all understood what is cortical nephron and what is juxtamedullary nephrons? Yes? Yes, Bhuvana, hello, hi, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Yes, I have changed it. Okay, Leela? Okay, fine. Yes, so cortical nephrons and juxtamedullary nephrons, is it fine? Okay, so now shall we move further? Yes. So we have this efferent arteriole, right? So this efferent arteriole, so it carries blood away from the glomerulus, right? So this carries blood away from your glomerulus. So this forms a minute network of tubules. So this forms a minute network of capillaries. Sorry, this forms a fine network of capillaries around your renal tubules. So these capillaries are known as peritubular capillaries. So these are known as peritubular capillaries, right? So this efferent arteriole which carries blood away from the glomerulus, so this forms a fine network of capillaries around your renal tubules. These are all renal tubules around your renal tubules, and they are known as peritubular capillaries. Okay, so these peritubular capillaries, okay, so these peritubular capillaries, they have a minute network of blood vessel. They have a minute vessel of blood capillaries or blood vessel, okay, which is parallel to your loop of Henle, which is parallel to your loop of Henle, and it is known as vasa recta. So, it forms a network of capillary around your renal tubule, right? So, that is your peritubular capillaries, right? So, one among the peritubular capillary, it forms a minute network of vessel which is U-shaped and it runs parallel to the loop of Henle. So, this is your vasa recta. Right? Is this clear? Yes? Okay, so you like handwritten notes, you like writing on the board, so that is more better. So you guys don't prefer text on the slide, but you prefer handwritten notes. That's nice. Even I love writing and studying because that makes sure you remember the concept for a longer time. Okay, so is this clear? So with this, I am done with the structure of kidney completely. So no doubt in the structure of kidney, you should never forget, right? So one day I will bring a session where I will explain the passage of digestive system, the passage of breathing, the structure of heart, the structure of kidney, then we will talk about the structure of muscle, the structure of brain, eye, ear, and finally the endocrine system. So all the major functions, all the major structures we will talk about. Okay? Yes? So is this done? Is this done, everyone? So no one will ever make a mistake in the structure of kidney? You should never, okay? Right. So now we move further. So now we come to the process of urine formation, right? So I will just give you an introduction because already it's going to be 5:45. So just I will give you an introduction. So how is urine formation done? So urine formation is done by your kidney. So we all know this much. So it happens in 3 processes. So the first one is glomerular filtration. Then you have reabsorption and then you have secretion. So these are the 3 steps. First is glomerular filtration. So it has 3 layers through which filtration happens. So you have glomerulus, then you have Bowman's capsule, right? So glomerulus, so it includes the blood vessel of your glomerulus, then you have epithelium of the Bowman's capsule and the basement membrane between the two. So all of these help in filtration. So the epithelial cells of Bowman's capsule, okay, so they are known as podocytes. They are arranged so intricately that they have filters or spores. So they can filter perfectly. They filter your blood, okay. Yes, we will talk about vasa recta. Vasa recta helps in the concentration of urine. Okay. Okay, Satyendra, your suggestion is taken, right? So these are known as filtration slits or pores. They are very, very accurate that everything passes. In the blood, your blood cells are neglected, your plasma proteins are also neglected, okay, only the plasma flows down. So this is known as ultrafiltration. So you have your glomerulus, you have your Bowman's capsule, so this is your Malpighian part, right? So we are talking about urine formation. Right? So in urine formation, you have 3 stages. One is glomerular filtration. The next one is reabsorption. Then you have secretion. So first we are talking about glomerular filtration, okay? Yes, okay, your suggestion is all taken. I will try to do mock test, okay? Right. So we are talking about your glomerular filtration now. So you have your glomerulus, you have your Bowman's capsule, right? So here you have your afferent arteriole. So here you have your afferent arteriole. Right? So what are the layers that is involved in filtration? So first you have the blood vessel. So the layers which is involved in filtration. So, first your glomerulus is a network of capillaries, right? So, the blood vessel are known as endothelium. So, we say endothelium of your glomerulus. So, the epithelial tissues which are made up of blood vessels are known as endothelium. So, endothelium of glomerulus, right? And then you have epithelial cells of your Bowman's capsule which are known as podocytes, right? And in between these two you have a basement membrane. So, all these layers are involved in the filtration process, okay, right? Fine. Now, these podocytes are arranged so intricately that they have what is known as filtration slits or pores. So, your efferent arteriole brings in blood, okay, so this filters your blood. So, this filtration is so perfect that even your plasma proteins are retained. Even your plasma proteins are retained. So, your blood has plasma and your blood has formed elements, right? So, formed elements include your RBC, include your WBC, and it includes your platelets, right? So, plasma has plasma proteins and electrolytes. So, your plasma proteins are also retained. Such accurate is the filtration of the glomerulus, thus it is known as ultrafiltration. Thus, it is known as ultrafiltration, right? So, this is your first step in the process of urine formation. Is this clear, everyone? Yes? Yes? Okay, yes, I will take up your suggestion and I will think about it and I will let you know. Okay, Satyendra? So, is this clear? Yes, erythrocyte is also RBC. So, the first step in urine formation is clear, that is glomerular filtration. So, in the next lecture, I will talk about reabsorption, secretion and the mechanism of concentration of urine. So, these are two important topics. Then we should also talk about the functions of the tubules. So, what is the function of the three renal tubules? So, these three things we will talk about in the next lecture. Alright. So, please have a happy weekend. So, study well. I will know when the next mentee is. Alright. So, have a great weekend. I will meet you on Monday. So, Monday 5 o'clock we are continuing with evolution. So, we spoke till your comparative anatomy. So, we will go from there. Okay. Yes, so yes, hi Natasha. So, thank you for joining our session. So, please do like, share and subscribe. So, I will meet you on Monday. So, until then, if you have any doubts, so please join the Telegram link that's given below my description box. I will meet you on Monday. So, until then, this is me wishing you more and most. So, take care and bye-bye.